Well, thanks a lot, Peter, and thanks, Nitin, for the invitation to come here and talk to you about some of our recent work on the human microbiome. So what I want to do today is make a three-point argument to kind of illustrate why I think the microbiome is really important. So I can make this argument in three different sections. Okay, first, I'm going to convince you that the microbiome reflects the state of the human host, and I'll talk to you about how we're sort of exploiting this property to, develop new medical diagnostics based on the microbiome. Second, I'll show you some evidence that diet and environment, lifestyle, can actually affect the composition of the microbiome. Okay, and then finally, I want to close the loop and show you that what's in our microbiome actually turns around and affects the health of the human. So you can imagine we put all these things together. You can imagine new treatments based on diet, environment, lifestyle changes that don't directly affect the host, but actually indirectly affect the host by modulating the microbes. Okay, so the first argument is this sort of vignette is from MD PhD student of mine in collaboration with Athos Bousvaros at Children's Hospital in Boston, and sorry about that. Basically, Athos came to us and he said, we've got a problem. He runs a pediatric inflammatory bowel disease clinic in Children's Hospital, and for an adult around my age, IBD is not terribly difficult to diagnose. But when an 8-year-old comes in, with presenting with depression, weight loss, and GI distress, it's kind of a hard choice. Do you send them out for a colonoscopy or not, right? So what we really want is a accurate, non-invasive test, and so we thought, let's look at the microbiome. So what I'm showing you here, we collected about 105 samples from patients, healthy patients. Well, these are control patients, so they're actually patients who were referred to the clinic. So this is really the control set that we want to test against, who had GI troubles but non-inflammatory disease, and then ulcerative colitis patients and Crohn's disease patients. And what you can see in the blue basically indicates we have the bacteria, the different groups on the rows here. All of the columns are different patients, and the darker the blue color, the more of that particular bacteria that patient had. And the one thing I want you to notice on this graph is there's no one ideal biomarker. The closest thing to like an ideal biomarker for inflammatory bowel disease is E. coli here, because we really only see it in high abundance in our diseased patients. But if we were to base our decisions solely on E. coli, we would diagnose correctly, we'd have sensitivity of like 10 or 20%, okay? It would be absolutely terrible. And all of these other predictors are sort of equally bad. But what's neat is we can combine all of these weak predictors to actually get a fairly accurate diagnosis of disease. And this kind of cemented the idea that, it's not a single classical Koch's postulates type disease. It's really a collapse of the ecosystem, all these weak signals that come together to form disease. Okay, so that should convince you that the microbiome tells us something about the health of the host. What I want to talk to you now about is work done by me and a student of mine, Lawrence David, who's now a junior fellow at Harvard. And this is work showing how diet and environment can actually influence the microbiome. So what we wanted to do is we wanted to know, in advance, we didn't want to come up with any a priori assumptions about what affects the microbiome. So we wanted to, get some volunteers to collect every single aspect of their life, okay? Every time their mood changed, we want them to record every morsel of food they ate, how much sleep they got, if they exercised, okay, over 300 different variables about their day-to-day life in real time. And then we wanted to also collect their stool samples every day, okay? Surprisingly, nobody actually signed up for the study. But we were able to find a determined graduate student and young assistant professor at the time who were willing to do it. Actually, my student said, hey, if I write an iPhone app so we can record this stuff in real time, can I charge it to the grant? So now, yes, there's an app for that. Okay. So this is my microbiome in 2009. Okay, and the first thing I want you to notice when you compare this to my students' microbiome is you could pick any day from my time series and any day from my students' time series, and you would know sort of beyond a shadow of a doubt which sample that came from. Okay, so these are two very, very distinct microbiomes. And the other thing I want you to notice is that the day-to-day variation is about the same as the month-to-month, week-to-week, year-to-year variation. Okay, so the microbiome is surprisingly stable, and you know, there's variation day-to-day, but over long periods of time, we see the same bugs again and again and again. But from each person to each person, it's actually quite different. Okay. Okay, so we recorded all this data. What actually, you know, correlated with the microbiome? Well, there were some dietary factors that influence the microbiome. So what you see here is a group of bacteria that sort of respond positively to fiber. But overall, there were a surprisingly small influence of diet and other events on the microbiome. So what did change the microbiome? Well, what I'm showing you here is a horizon plot. Okay, so this is the beginning of our study. This is the end of our study. Okay, and the rows here are all the different bacteria. The warm colors, like red, indicate there was more of that particular bacteria on that particular day, and the blue colors indicate there's less of that bacteria on that particular day. So about halfway through the study, my student's wife got this great internship in Bangkok, and he asked me, oh, hey, can I, you know, take the summer off and go hang out in Thailand with her? And I said, well, okay, I'll relieve you from your duties as a graduate student, but not as your duties as a study participant, okay? Which was a good idea in retrospect, because you can see what's really neat is this is while he was in Southeast Asia. So you can see, this is what I'm talking about, the stability of the microbiome, but as soon as he got off the plane to Southeast Asia, he came down with a cocktail of 3 pretty nasty enteric pathogens, and they really took over, and they drove out all his normal commensal flora. But almost the day he got back off the plane to the United States, pathogens were gone, and his microbiome was completely back to normal. Okay? All right, a little bit later in the study, a friend of mine invited me and my son out to brunch with him and his young daughter, and I made the mistake of ordering the special Greek French toast, okay? So about 48 hours later, I was deep in the throes of food poisoning. I went to the doctor said, oh, it's a virus, you don't need antibiotics, so I now have DNA evidence. He was wrong. But that's okay. It's okay, because this is actually some of the best data we got in the study. So you can see, so these are, this is salmonella down here. And what's really interesting is as soon as the salmonella takes over, all of these bacteria basically are depleted. Now, a few of them, after the salmonella infection, come back. But you can see the majority of the stuff that sort of, got depleted went completely extinct. And these guys basically took over. So about 30 to 40% of all the bacteria went completely extinct and got replaced by entirely new species in a matter of days. And what's really interesting is now these species, this is the normal flora in my gut, and these are very, very stable and resilient over time. Okay? Okay, so hopefully I've convinced you now that the microbiome reads out the state of health of the human. Diet and environment can influence the microbiome. What I want to talk to you now about is sort of closing that loop. The microbiome can then feedback and affect the health of the host. So this is all work done in collaboration with Susan Erdman, who's a wonderful collaborator in MIT's Department of Comparative Medicine. And we really got inspired by this paper that came out in New England Journal of Medicine. The long story I won't get into, this work was actually truly inspired by a miscommunication between me and Rob Knight, which I can tell you about later if you're interested. But part of the motivation was this New England Journal of Medicine paper where they went through 1.6 million years of patient data to identify the factors that lead to long-term weight gain and obesity. Some of the unsurprising findings of this study were that potatoes, so like French fries, processed meats and sugary drinks were the worst thing you could do in terms of long-term weight gain, so what we're calling the fast food diet. But a real surprise here was that yogurt. So yogurt was sort of the best thing that you could eat to maintain long-term lower weight. And this was kind of surprising because the yogurt didn't match the profile of the type of foods that, led to maintaining a lean body mass, okay? Because this is just folks coming in every few years and saying, checking a box in terms of how much yogurt they eat. So this is not some super health food yogurt. This is the average yogurt people are eating. It's probably laden down with sugar. But what was really surprising is for every additional serving of yogurt that you ate per day, you not only did you not gain weight, but you lost 1.5 pounds per 4 years. Okay? So we said, in the discussion of this paper, they say, well, it doesn't really match a lot of the other foods, so maybe it has something to do with the microbiome. So this is really exciting to us. We started a mouse study. We fed mice fast food. We fed them yogurt, and we also had a study going on at the time with David Hafler over at Yale, and a lot of his patients, just by chance, had been eating a lot of fast food. So one of the things we noticed is that we could tell that we could detect the signal for the fast food diet in both the humans and the mice. And in the mice, when we looked at body weights, whether they were on the control diet and got yogurt, or they were on the westernized diet and got yogurt, they always had lower body weights when they were given the yogurt. Now, at this point, we really hadn't gone too much farther than the original study because we still didn't know, is it really the probiotic bacteria that's making a difference? Or is it something about the dairy or vitamin A, vitamin D, usually enriched in yogurt? So what we did is we purified just the Lactobacillus reuteri and we stuck it in their drinking water, okay? And what we found is even more dramatic effects in terms of weight loss, okay, whether they're on the control diet or the fast food diet. And then what was really surprising, I don't know if you can see these images super well. So here's our 9-month-old Swiss mice eating fast food every day. And then here's our 9-month-old Swiss mice eating fast food, but also getting yogurt bacteria in their drinking water. Okay, so they're skinny. Yeah, you can sort of tell them side by side here. But they were also like really, really shiny. Okay, so my collaborator wrote to me because our tech was out of town on vacation. She had to go and harvest the mice herself, and she's like, in 2 decades of harvesting these mice, I've never seen mice like quite this shiny. And then she started thinking, so she's got this dog, Julio. So Julio is here, and you can kind of see he's got this dog wound, this dog fight wound over here. So she had been feeding him probiotic Stonyfield yogurt every day in hopes that it would help his wound healing. And she said, everybody who comes to my house says Julio is the shiniest dog they've ever seen. So logically, there must be a connection between eating yogurt and having a shiny coat. She's got this other dog, I don't know if you can see here, Zappy. It's nowhere near as shiny as Julio. It's not really rendering well here. Maybe you can see it on the monitors. So she said, we should feed Zappy Stonyfield yogurt every day. And so this is Zappy today. Now, arguably the shiniest dog on the planet. So I know what you're thinking. These guys at MIT do pretty rigorous science. But in the meantime, while we were doing our dog experiments, we had sent off the histological samples from those original mice. And what was really surprising, we didn't have a lot of hope here that we'd see something really obvious because hair histology is pretty complicated. But even to the untrained eye, you can see here a huge difference between the mice that were on a controlled diet and the mice getting Activia. Okay, so here you can see those actively growing hair follicles in anagen phase, whereas the normal control mice, the hair follicles were mostly intelligent phase, totally keratinized and dead, not actively growing. Okay, so the backstory here is that our histologist was working in Greece at the time, during the time of the financial crisis, so it actually took a really, really long time to get these samples because he had to actually climb a fence and pick a lock to get in because his institute was not open during the strikes. So, you know, this is part of the reason we resorted to the dog experiments in the meantime. Okay, so I think I made my 3-point argument, okay? The microbiome reflects the state of the host. Diet and environment affect the state of the microbiome, and the microbiome can also influence the state of the host, okay? What I want to do is go back now really quickly and show you that diet and environment can alter the microbiome, but in a very unexpected way. So this was really motivated by this beautiful paper that came out in Nature in 2010 that in my lab we call the sushi gene paper. So if you guys are not familiar with this, it was this really great story where they went out to look at these carbohydrate metabolizing enzymes of these algal polysaccharides that have a lot of sulfur group modifications. And so these are, you know, very specific enzymes that can metabolize these polysaccharides. So they were solving the crystal structure, and they blasted for homologs. They found a lot of homologs in marine bacteria, and they concluded that the marine bacteria had stolen this gene from the algae that's making the polysaccharide so that they could use it to eat that algae. Okay, it was kind of a neat story. But then the other thing they noticed is they found it in 4 species of Bacteroides, okay, that were originally isolated from the human gut. And they're like, what is it doing there? Well, in fact, it was isolated from the gut of 4 Japanese individuals. And they said, you know, Japanese people actually do eat a lot of this particular species of algae because it's the exact same species that's used in those nori sushi wrappers, okay? So they went to metagenomic library, they looked for this gene, they actually found it was quite common in a metagenomic library from Japanese people's guts. They went back to even larger database taken from North Americans' guts, and they didn't find a single copy of the gene. So they concluded that at some point, a bacteria probably feeding on the algae got into a piece of food that somebody ate, and the gene from that bacteria transferred into the gut microbes of that person. And I thought this was great. And I thought, you know, we should take all the genomes that are out there, and we should just do a search and see if we can find like 5 or 10 more examples of like really cool anecdotes like this. You know, where we could find basically a smoking gun, right? 100% identical DNA into totally different species. We could find just a few examples of that. It would be really cool. So my students came back about a week later, and they said, Eric, we've got 80,000 examples of this, okay, and like 11,000 unique proteins. They were rotation students, so naturally I didn't believe them. But I got a more senior graduate student in the lab to do the experiment, and yeah, we got the same result. But what was even more surprising is that almost every example that we found where there was 100% identical DNA in 2 radically different species, both of the bacteria were isolated from the human body, okay? So here's the map of basically all the bacteria that we looked at, basically all the sequenced bacteria at the time. So about 1,000 bacteria from the human body and 1,000 bacteria from all other environments. And what you can see is I'm showing you the rate of horizontal transfer between bacteria with, you know, this particular annotation. So you can see down the diagonal. So these are horizontal transfers, very recent horizontal transfers between bacteria in the same environment was always more common than transfer to any other environment. And you can also see the sort of block over here. This is all transfer within the human body, but even within the human body, transfer was much more likely if the 2 bacteria were isolated from the same site on the human body. Okay. Now let me just clarify. This isn't 2 bacteria from the same human body. As far as we know, none of these bacteria were actually isolated from the same person. But this is like 2 bacteria, either both from the gut of 2 different people, or both from the airways of 2 different people, or both from the skin of 2 different people. Okay, and the same trend held for the non-human environments. It was just a much weaker signal. Okay. All right, so one of the things we were really actively interested in is what about horizontal transfer of antibiotic resistance genes, okay? So what I'm showing you here is now the bottom half of this plot is showing you for all the transfers that went on, what fraction of those encode at least one antibiotic resistance gene, okay? But actually, if we look back up here, at where we see horizontal transfer, I said, mainly it's between bugs and the human body, but we also see an excess of horizontal transfer between bacteria isolated from human foods or from farm animals and the human body. And if we turn to the other side of this plot, we see that these particular off-diagonal transfers to and from the human body in our food and our livestock, actually more than 60% of them include at least one antibiotic resistance gene. So it tells us that we really have to think carefully about some things like our subtherapeutic antibiotic treatments, because even if the microbes that are antibiotic resistant don't thrive in the human gut, we're showing that their genes can actually freely move between themselves and bacteria that do thrive in our guts, okay? And I will stop there and be happy to, no, I won't. I will not be happy to take questions for a little while.